Okay, next is a carbohydrates. Carbohydrates serve as a fuels and also as a building materials. Okay, what is a carbohydrates? Carbohydrates includes of sugar and a polymer of sugar. The simplest carbohydrates consist of monosaccharide. What is a disaccharide? Disaccharide is a double sugar, consists of two monosaccharide joined by covalent bond. Carbohydrates, macromolecule, are polymer called polysaccharide composed of many sugar building blocks. Okay, look into this slide. A carbohydrate consists of four groups of carbohydrate. The first one is a monosaccharide, known as the simpler sugar. Next is a disaccharide, as I mentioned before, consists of two monosaccharide joined by covalent bond. Okay, next is an oligosaccharide. This oligosaccharide may be new for you. They consist of 3 to 14 monosaccharide. And the last one is a polysaccharide. It's a polymer composed of many sugar monomer. Monosaccharide generally have a molecular formula. There are some multiple of unit CH2O. Glucose, known as a C6H12O6, is the most common monosaccharide and very important in chemistry of life. In the structure of glucose, we can see the trademarks of a sugar. The molecule has a carbonyl group CO and a multiple hydroxyl group OH. Refer to this slide. We are shown example of glyceraldehyde. Glyceraldehyde consists of 3 carbon sugar and other groups of aldose or aldehyde sugar. They are hydroxyacetone is a 3 carbon sugar isomer for glyceraldehyde are the groups of ketose or ketone sugar. Okay, this is a difference between aldose and ketose group. We look at a pentose of 5 carbon sugar, ribulose and ribose. This ribose and ribulose we will discuss later in nucleic acid part. What is the difference between ribose and ribulose? They are location of carbonyl group. For carbonyl group, at the carbon number 1 for ribose and carbonyl group at carbon number 2 for ribulose. Okay, now we look at difference between aldose and ketose. This is an example. Glucose and galactose is an example of aldose, and fructose is an isomer of glucose is a ketose. So this glucose and galactose difference compared to glucose at carbon number 2, which is glucose and galactose, we have carbonyl group at carbon number 1 compared to fructose, the carbonyl group at carbon number 2. And the difference between glucose and galactose at carbon number 4 is the location of hydroxyl. Still, another source of diversity for simple sugar is the special arrangement of the parts around asymmetric carbons. Glucose and galactose, for example, differ only in placement of parts around asymmetric carbons. This small difference is significant enough to give the two sugars distinctive shapes and binding activities, thus a different behavior. Okay, we discuss on monosaccharide. Monosaccharide is particularly glucose, major nutrients for cell. In the process known as a cellular respiration, cells extract energy from glucose molecules by breaking them down in a series of fractions. Not only a simple sugar molecule, a major fuel for cellular works, but their carbon skeleton also serve as a raw material for the synthesis of other types of small organic molecules such as amino acids and fatty acids. Monosaccharides has a few properties like sweet teas and also can be crystallized. This is because of polar due to hydroxyl group attached to carbon atom and form hydrogen bond with water. This monosaccharide easily dissolve in water and have a reducing properties. Okay. For monosaccharide, the ring from glucose exists in the form of alpha and beta glucose. What is the difference between alpha and beta glucose? And alpha glucose, the location of hydroxyl at carbon number 1 is below the plane of the ring compared to beta glucose. At carbon number 1, the hydroxyl group is above the plane of the ring. Um, when the alpha glucose molecule are joined in chemically to form a polymer, starch is formed. But when a beta glucose molecule are joined to form a polymer, cellulose is formed. The monosaccharides are usually drawn as a linear skeleton. The chemical structure equilibrium between a linear and ring structure greatly favor the form of ring, which is the ring structure are more stable. The carbon of sugar are number 1 to number 6, as shown as this figure. Look at the glucose carbon number 1, the magenta color, bond to the oxygen with the blue color attached to carbon number 5. Disaccharide consists of two monosaccharides joined by glycosidic linkage or glycosidic bond, a covalent bond formed between two monosaccharides by dehydration process. These disaccharides they have a character quite similar to monosaccharide, which is water soluble and sweet tasting and also can be crystallized. Okay, they have three examples of the saccharide: maltose, sucrose, and lactose. Okay, the first one, maltose, also known as a malt sugar. Okay, this maltose is a disaccharide formed by a linking of two molecules of glucose. This malt sugar is an ingredient used in brewing beer. Okay, the most prevalent disaccharide is sucrose, or you can find in sugarcane. Um, this sucrose, we also known as a table sugar. Okay, the is a two monomer, glucose and fructose. Plant generally transport carbohydrates from leaf to roots and other non-photosynthetic organ in the form of sucrose. We will discuss transport of sucrose in chapter 5. The last one is lactose. The sugar present in milk is another disaccharide. In this case, a glucose molecule joined to a galactose molecule. And last column show the character of disaccharide. Maltose and lactose are reducing sugar compared to sucrose is a non-reducing sugar. Okay, what is the difference between reducing and non-reducing sugar? In carbohydrate, which is capable of being oxidized and cause of reducing of other substances without having to be hydrolyzed as a reduced sugar, but those which are unable to be oxidized and do not reduce other substances are known as non-reducing sugar. Okay, generally all the free monosaccharide, okay, which is having free aldehyde or hydroxyl ketonic groups are capable of being oxidized. After being oxidized, they cause the reduction of other substance, and so they are known as a reducing sugar. Okay, we discuss detail in reducing sugar. Reducing sugar have a free aldehyde or ketones groups. This reducing sugar have capacity to reduce cupric ion in Benedict solution. Okay, this Benedict solution we will test in experiment 1 in lab session. Example, reducing sugar is maltose and lactose. Okay, this slide shows dehydration reaction in synthesis of maltose. Okay, as we discussed before, maltose is the binding between two monomer of glucose. This process known as a dehydration, which is the remove one molecule of water. Okay, the binding between these two monomers of glucose at carbon number 1 and carbon number 4. That's why the binding known as a 1,4-glycosidic linkage or 1,4-glycosidic bond. Okay, next, B is a dehydration reaction in synthesis of sucrose. This formation of sucrose is between two types of monomer, glucose and fructose. They are remove one molecule of water, the hydroxyl at carbon number 1 and hydrogen group at carbon number 2. So the linkage known as a 1,2-glycosidic linkage. Okay, the last one is formation of lactose between galactose and glucose. Okay, the binding remove one molecule of water, the hydroxyl group at carbon number 1, and the hydrogen group from carbon number 4. So that the bond is between carbon number 1 and carbon number 4, known as a 1,4-glycosidic bond. Oligosaccharide may contain 3 to 14 of monosaccharide. The link between the monomer may be involve 1,2-glycosidic bond or 1,4-glycosidic bond. Where we can find this oligosaccharide? Normally, we found in the surface of cell or the surface of plasma membrane. Example here is a glycoprotein and a glycolipid. What is a glycoprotein and glycolipid? We will discuss detail in chapter 2. Act as a cell recognition. This glycoprotein example we found in extracellular matrix of plasma membranes.